So in order just to understand linkage, we have to remember back to some stuff that Mendel did. So remember, one of his laws is the law of independent assortment. And this was the idea that traits are inherited independently from each other. And so in the example we looked at in the previous chapter, we saw, for example, that you could get new combinations like yellow seed color with wrinkled seed shape that were not seen in the previous generation. So in the previous generation, the parents had been yellow round and green wrinkled. Yellow wrinkled and green round were both new combinations that appear because the traits are inherited independently from each other. They don't have to be passed on in the combinations that the parents had. So when you're trying to understand linkage, make sure you go back and look at that law of independent assortment if you didn't quite understand it before. So what Mendel didn't know at the time was that independent assortment, his law, is only going to apply to genes on different chromosomes. Why is he not going to know that? Well, remember, Mendel is doing this in the 1800s, long before we know that DNA is the genetic material and that chromosomes are the genetic material. So what we now know is that if genes are close together on the same chromosome, then they're what's called linked, and this is linkage. So they are linked genes. They will not follow the law of independent assortment. And they will tend to be inherited together in the same combination of traits that the parents had. So they violate the law of independent assortment. One example where they were very surprised to see the law of independent assortment not occurring was when these guys, Bateson and Punnett, were crossing sweet peas for flower color and pollen shape. So again, sweet peas are related to garden peas, but they're going to do stuff a little bit different. So they were going to do a dihybrid cross. And again, remember that at the end of a dihybrid cross, you should expect to see a 9 to 3 to 3 to 1 ratio. But instead of seeing a 9 to 3 to 3 to 1 ratio, they saw much higher rates of parental phenotypes. So that's more of these and more of these than they would have been expecting. And so because they see more of the parental phenotypes, remember that's how they came from the parents, not new combinations, it suggested that the traits were linked. So that's the cross that they did. So again, the dihybrid cross, purple flowers long pollen, red flowers round pollen. So they get the dihybrid, and like they were expecting, they see the dominant traits. And then they do a self-fertilization, and this is where they're expecting the 9 to 3 to 3 to 1 ratio, because that's what Mendel saw in garden peas. But instead, they see about 16 to 1 to 1. I'm rounding here. I guess I should. I'm trying to round in my head. That's not working out very well. So they see 15.6 to 1 to 1.4 to 4.5. So like I said earlier, they see way more of these and these. They would have expected only 9 and 1 of that, but they see much more of those and way less of these. And these are the parental types. So purple flowers and long pollen, this is also purple flowered and long pollen. And red flowers round pollen is also this one. So that's why they're called the parental type. The new combinations, purple flowers with round pollen, that you don't see in previous generations, there's not very many of. And so again, their conclusion is that these traits are linked. They do not follow the law of Mendel of independent assortment that Mendel came up with. So the person who most understood and studied linkage was a guy called Thomas Hunt Morgan, who was working on genetics in the early 1900s. And he focused not on plants, but on the fruit fly. And so he looked at traits in the fruit flies, and just to give you a little background about fruit flies, they use a totally different lettering system, of course, sorry, than we use in plants. So they're going to use these plus for the dominant or wild type allele, and not a plus for the mutant or recessive allele. And then they use the letter of the recessive trait. So you're not going to use these that much. It's just to help you understand this figure. So we start out with a homozygous dominant for gray body and straight wings, and she's crossed with a homozygous recessive. And so what you get is a hybrid, a dihybrid female. So she's heterozygous for the traits, and then she's crossed with another homozygous recessive. So this is not a dihybrid cross. Okay, so you're not expecting a 9:3 to 3:1 ratio. Because she's being crossed with this guy, what you would expect is a 1:1 to 1:1 ratio. So if this was following the law of independent assortment, you'd expect to see a 1:1 to 1:1 to come out of this cross. But instead, what he sees is a much greater amount of these two phenotypes. So just to give you an approximation, instead of a 1:1 to 1:1, they see about a 3:3 to 1:1 ratio. So way more of these two. And if you look here, gray body, straight wings, oh look, that's what the mom had, right? And black body, curved wings, oh look, that's what the dad had. And the dad had here. So those are what's called the parental types. And so they found way more of the parental types. They still had a small number of these ones. There weren't very many of them. The gray body with the curved wing, the black body with the straight wings, these are called the recombinant or non-parental types. They're the new combination of traits. So again, his conclusion is that the traits are linked. They are on the same chromosome. So if we look at the chromosomes here, B plus and C plus are next to each other on the red chromosome. B and C are next to each other on the blue chromosome. So they're going to travel together because of how meiosis occurs. Meiosis separates chromosomes. So the parental offspring come from that B plus and that C plus being on the intact red chromosome, and the B and C being on the intact BC chromosome. Now notice that the recombinants, there are not as many of them, but notice this. This is what I'm going to highlight here. This is now a hybrid chromosome, so mostly red with a little bit of blue, and this one is mostly blue with a little bit of red. So the recombinants are a product of crossover during meiosis. So you mostly get non-recombinant or parental offspring because the traits are linked on the same chromosome. You do get a small number of recombinant or non-parental offspring because of crossover during meiosis. So Thomas Hunt Morgan made 3 proposals, and just summarize them here. When genes are on the same chromosome, they're not going to follow the law of independent assortment that Mendel had. The genes are going to tend to be inherited together, and that's where the parental type offspring come from. Crossover will occur during meiosis, where the homologous chromosomes, so just to remind you, the homologous chromosomes will exchange pieces. When these exchanged pieces become part of the gamete and get fertilized, that'll lead to non-parental type offspring, or the recombinant offspring. His third, and one thing that he used a lot, his proposal, he said that the likelihood of crossover is going to depend on the distance between 2 genes. There's more likely to be crossover when 2 genes are farther apart. So the greater the distance between the 2 genes, the greater the number of crossover events, and the more crossover you have, the more recombinant offspring you have. So there's a relationship here. The farther apart they are on the chromosome, the more crossover you'll have, the more recombinant offspring will be present. So he used this principle to develop things called genetic linkage maps. So he would carry out test crosses and measure the rate of crossover by looking at the number of recombinant offspring. And he was able to map the fruit fly genome. So remember, this is before we know about DNA. We don't even know that DNA is a genetic material. But Thomas Hunt Morgan and his colleagues made maps of the fruit fly chromosomes. They just basically established an endpoint, which was 0, and then made map units that were based on crossover to give relative distances between the genes. So this is a question to just think about how they did this. Which would show more recombinant offspring, a cross with dumpy wings and brown eyes, or a cross with dumpy wings and black body? So remember, there's a relationship between crossover, distance, and recombinant offspring. So the greater the distance, the greater the number of crossover, the greater number of recombinant offspring. So if I look at dumpy wings and brown eyes and compare that with dumpy wings and black body, these two are further apart. Therefore, I would expect a greater number of recombinant offspring between dumpy wings and brown eyes. Again, because the greater the distance, the greater the number of crossover, the greater number of recombinant offspring. And so that's how Thomas Hunt Morgan established these genetic linkage maps.